Hi, Ainsley. Thanks for joining me today. How are you?
I'm good. How are you?
Good. That's great. We had met at
hmm
ACS
Yes.
2 weeks ago in Washington, D.C., and I'm excited for you to be here and tell us about your work. So I was hoping you could introduce your work on hemoglobin variant testing.
Yeah, for sure. The reason why we even did this research in the first place was that we found that there was like a kind of lack in the way that people usually test for hemoglobinopathy. The main background for this is that hemoglobin is a very big protein. It's composed of four subunits, and each of those subunits is multiple amino acids long, which means that if one amino acid goes awry, then the whole protein is a bit distorted. The implications for this is that when the protein gets distorted, there can be a lot of diseases or pathologies that can ensue. For example, one very popular one would be anemia and sickle cell disease. And one niche that we specifically focused on was detecting and identifying those that are very close to their original subunits by mass because we're using a mass spectrometry method. Because we're using a high resolution mass spectrometry method, our method allows us to resolve those subunits that are like 1 Dalton or even less than 1 Dalton away from the original subunit.
Then you just touched on the mass spectrometry.
Mhm.
Your team decided to use the LC high resolution mass spec, and I was curious why you used LCHRMS over other well established tools, possibly like capillary electrophoresis or gene sequencing.
Mhm. So our goals were mainly to first be able to both identify like the presence of a variant and like the identity of the variant itself. And then we also had to make sure that this method was cost efficient or relatively cost efficient, time efficient, and requires like low sample preparation is the main thing. And so the thing with gene sequencing is that it's great because of its high accuracy, but it requires so much manual sample preparation and it requires so much time and cost to run. So it's really just not super feasible for daily clinical use. And then capillary electrophoresis, it does have the ability to separate the subunits so we can determine like the signature peaks of when these subunits separate. But also, it doesn't allow for that high resolution power or high resolving power that we see with high resolution mass spec. And that's, I think, the crucial point here is that we're trying to identify what these subunits are, what exactly they cause in the human body. So we have to really hone in on that accuracy.
Makes sense. One thing that stood out was your use of the C4 reverse phase column for the
Mm-hmm.
separation,
Mm-hmm.
which is different than what some people might consider the more common C18 column. So, what advantages did that allow?
Okay. Yeah, so I think the main thing with the C4 column is that we actually did try it with the C18 column. It did not work, so that is why we opted for the C4 column. And I think the reason is that the C4 column is just like it has a smaller carbon chain, which means that there's wider pores in the column and there's less of a chance of the protein getting retained in the column itself or kind of denatured by just getting stuck in the pores. And with the C4 column, we saw much better elution overall.
If someone was to try to replicate some of this work, what are the most important or key parameters, whether that's temperature, gradient, resolution, that you'd really emphasize for reproducibility?
Okay. So for sure the C4 column, I would say that is really important. And then I would say probably the mobile phases. It's important to keep those the same. So for our experiment, we used two mobile phases. One was I think 0.1% formic acid in water, and then the other was 0.1% formic acid in acetonitrile. And then we just varied the concentrations of those as the chromatography progressed. And then, yeah, I think that also something important to note is that the temperature of the chromatogram has to be consistent. So I think that for ours we used 60 degrees Celsius. And then since we also like our experiment used a LC coupled with mass spec, we kept the capillary temperature of I think 320 degrees Celsius.
One of the notes was that mass spec isn't as widely adopted in clinical labs
Mhm.
compared to standard HPLC or CE. What do you see as the biggest barriers to implementation of mass spec in this area?
Okay. I think that one of the big barriers is that mass spec is still relatively energy high. So just the cost of maintaining a vacuum for the mass spec is quite high. So our lab had to have special conditions in order to maintain that vacuum. And I feel like it's just not super feasible in terms of space, energy. But I feel like I have seen certain improvements in the mass spec world. For example, this summer I was doing research at Purdue and I saw that there was this mini mass spec that people use for the same purposes as a regular mass spec. So I've never heard of people coupling a mini mass spec with LC, and I don't know how high the resolving power is, but I feel like there could be room to play with that. I feel like, yeah.
That's fair. Were there any unexpected findings or aha moments that were especially revealing to you while you were doing this work?
So I feel like, well, there's a big one, and that big one is just the C4 column because we tried so long with the C18 column to make it work, but in the end, we found that the C4 column worked so much better. And then I would say another moment was it's less surprising, I think, but it still just goes to prove how high the resolving power of our method is. So basically, we had two types of samples. So we had adult samples, which I think is just anyone above the age of 2 is an adult. And then we had people below the age of 2 who are considered neonates. And the difference between these two groups is that neonates have a different subunit structure compared to adults. So adults are high in hemoglobin beta, or like the beta subunit, whereas neonates have this subunit in very low quantities. But with our sample, our neonate did have a variant on the beta subunit, and our mass spec was still able to detect that variant despite it barely showing up in the chromatogram. So that was a really surprising moment for me.
Well, as you said earlier, I think everyone would have started with that standard C18
Yeah.
column. You ultimately navigated to the best approach.
Mm-hmm.
One thing that stood out about your research is you're at an interesting intersection, let's say, between analytical chemistry and clinical medicine
Mm-hmm.
almost. What excites you about this type of work in the future as it relates to proteoform analysis?
I feel like there's so much room to explore, and I feel like it's not just about hemoglobinopathy, because as you said, proteoform analysis can extend to so many things. For example, I've recently seen people use mass spec for cancer detection. It's in the works, of course, but the basic methodology is that they sample healthy tissue and unhealthy tissue or tumorous tissue, and then they compare how the mass signatures differ. Using that, they can create a calibration of the signatures that would determine unhealthy tissue versus healthy tissue. The point of this is to hopefully one day expand it into a clinical setting, just like our research. It's very exciting stuff.
Absolutely. For clinicians or laboratory techs who aren't as familiar with high resolution mass spec, what would you say to convince them in your type of work or testing that this approach has a lot of value? How would you
Mm.
kind of sell this approach to people who could benefit from it?
I would say that it's more about the potential at this point rather than the actual, what we see with our eyes. Because, well, what we see with our eyes is that this method is really accurate. It has a high resolving power. It's also just, it's good for those specific, detecting those specific niche variants. And that alone, I think, is a very key feature that not a lot of methods have. And I think the potential for this is that, one day we hope to expand it into a clinical setting. So we do have to tamper with how much energy is required to really maintain this instrumentation. But I think it's just a matter of time. I think really, if people are inclined to invest in this kind of research, like it can happen.
What would you say is the most important takeaway that you'd like the listeners to understand about your work?
I feel like the implications are actually pretty big. So if you think about it, analytical chemistry is really just like measuring or quantifying substances. It's not just about how specific we can get this measurement to be. It's really about how can we expand these techniques to other fields. So for example, biology, environment, energy. I feel like there's so many intersections of analytical chemistry with other, other sciences. And I feel like, for example, in the energy sector, you can use material sciences to create substances that do help the environment or like help trap greenhouse gases. And I don't know, I feel like it just goes to show how versatile analytical chemistry is.
The question I usually like to ask at the end of the interview relates to any advice that you might have, Ainsley, for other young chemists. And certainly, if you'd like to talk about that generally, if you have advice, I'd love to hear it. But it might also be interesting for you to talk about your recent ACS experience, because I don't know if that was your first poster presentation in front of such a large audience. Did you have any thoughts on how that went or advice to others who are doing that for the first time?
So that was actually my first poster presentation. You're right about that. And it was actually my first time, being involved in such a big project. And I think the general advice I would have there is if you do want to get involved in a big project at a young age, don't feel afraid to reach out to professors or researchers. They're usually very supportive. And because they've also come from that early stage of being a young scientist, not knowing who to reach out to, they're usually very supportive and they'll likely take you under their wing if they have the time and space. But yeah, just don't be afraid to reach out. And when you do arrive at your internship or your research opportunity, don't be afraid to reach out to the adults around you to ask them, what am I doing? Am I doing this right? And then also just pivoting to my poster presentation at ACS, when something like that happens, like when you do get an opportunity to present your research, the same thing. Don't be afraid to ask for help from the adults around you in the scientific community, because my poster presentation itself, I probably took the input of like at least 3 or 4 people in my lab. So yeah, it really just comes down to how willing are you to ask for help, because in the end, scientific research isn't like a subjective endeavor, or rather not like an independent endeavor. It's more like a group activity, a group effort. So really just make those connections and just get yourself out there.
That's a great answer. The need for teamwork is a topic that's come up a bunch on this forum, as you might
Mm-hmm.
expect. I think I just wanted to mention that I think the willingness to help other people and the willingness to share research is, in my small part of the world, part of what's facilitated these conversations, because
Mm,
people have been generally interested, whether it's current students or professors, willing to come on and share their work because they're excited about it and they want to help other people. They also see the important teamwork aspect of that, even beyond the lab, in terms of researchers in different places who are working on similar projects. And doing these interviews has really opened my eyes to all of the inter-school projects, because sometimes you see a lot of presentations and there might only be one institution listed, but I've
mm-hmm.
been able to talk with a lot of researchers where they're in constant partnership with other organizations, whether it's schools or commercial
Hmm.
laboratories, people in industry. So there's a lot of teamwork that I think goes unseen almost.
I think that's also like a reason that you went to ACS, right? Just to see how many people are presenting on different things, especially in the LC world.
Absolutely. It's one of the best ways to stay up to date with what's going on and
Yeah.
learn about what's coming next as well. And
Hmm.
certainly in my area of focus with more LC, MS, GCMS, it's always interesting to seeing the challenges that people have, specifically in sample preparation and where
That's true.
my company's products might be able to assist, but even uncovering new problems that we're not aware of that we might be able to help with in the future. It's definitely
Hmm.
important to
Yeah.
research and development.
Good market research.
Yeah, absolutely. Well, Ainsley, I really appreciate the time today. Thanks so much for joining me, and I hope to run into you at another conference in the future.
Yeah, for sure. Thanks for having me.